Okay. Hello, my name is Panhavuth, and this is my research project for the summer of 2022. So I'm looking at the resin preference of solitary bees. Now, why should we study native bees and specifically resin as well? So, so the reason why we're interested is simply because the fact that there's not much knowledge on native bees and resin in general. So there have been study in the areas. There have been study in regard to bees, but a lot of the resource have been put into studying honeybees as opposed to native bees. That being said, both native bees and honeybee has seen a significant decline in their population, and yet the general public only really seems to know about honeybees. Now there are many reasons for this. Of course, much of the product that we consume is made by honeybees, so of course we have more regular contact with them. But that being said, native bee is still very important, and we should still dedicate resources to studying them. So in a study by Kopek and Burt 2017, so the data that they got themselves, they saw that 24% of the native bees that they study are in peril, and that there's a population decline are occurring in 52% of the native bees that they study. Now, in a study before this, so in that same report, they also compile the data of studies before this as well. So this is the conservation. If you look on the right figure over here, so this is the conservation status of 4,337 North American and Hawaiian native bees. And this is taken from the IUCN Bumblebee Specialist Group, the NatureServe, and the Xerces Society. And as you can see here, 7.3% of the bees that they looked at, they were able to determine the status of. And of that 7.3%, they found that 58% is vulnerable to extinction. So this is a problem. And so what are the reasons for this? So the main cause of this is because of shrinkage in their habitat. So they're not able to find the appropriate resource for nesting, and they're not able to find the floral opportunity is of course shrinking. So they're not able to get the resource that they need to thrive. Now, there are many studies on the importance of flowers for bee, but not so much when it comes to resin and its usage by bees. So it's great to know that they have pollen and nectars, of course, but the problem would still exist if they don't have the material needed to make their homes. So that's why we're studying it. And of course, there's not much information, or at the very least, we were not able to find much information regarding resin preference by native bees in the literature. So what is resin? So first off, resin is a nonpolar substance. And resin can be defined as a mixture of compound of either terpenoid or phenolic origin. And it's produced by plants in the internal duct or on the surface gland. So as you can see here on the figure, on the top right figure over here, if you look at picture A, this is the transverse section of the stem of a young pine tree. And if you look at picture B over here, it actually shows the axial resin duct in the xylem and the cortex, and it's a close-up of the xylem resin duct. If you look at picture C, it's actually the cortical resin duct. And for picture D, you can see that it's a xylem axial resin duct in the annual growth ring of an adult Panhavuth tree. So this whole figure, they're all the internal structure, the internal resin structure of a tree. Now, if you look at the picture at the bottom right, this is actually a resinous leaf bud. So you can find this resinous leaf bud in temperate angiosperm. And as you can see here, there's this yellow substance that is dripping out. That's the resin. Now, if you were to look at conifer tree, if you see a wound and there's sticky substance gushing out of it, that's most likely the resin. So the terpene and phenolic are specialized metabolite, so they don't have like direct functioning for the growth and development of the plants. But resin plays an important role in the interaction of the plants and the environment. So what they do is that they serve to protect trees from pathogens and herbivore. So again, as I mentioned before, if you go up to a conifer tree and you see a large gushing wound out of it, if there's resin coming out of it, once that resin is exposed to air, that resin will harden. And when that resin hardens, it can also block the wound and stop harmful things from possibly harming the tree. And it also helps against herbivore as well. So um, so 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 resin have the ability to attract parasites of herbivore that can potentially harm the tree. Uh, and this kind of serve to and this act as like okay, I'm so sorry. So the resin itself can attract parasite and can attract creature uh, um, that kind of prey on the herbivore that possibly want to harm the tree. So the resin also serve to attract pollinator and seed dispersing animal via smell, color, and taste. So now we're going to look at resin usage by bee. So as you can see on the picture on the top right, this is a bee collecting resin. So resin is used in the construction of the nest. So the plasticity of the resin, it can be manipulated by bee for the formation of nest structures such as nest lining, the partitioning of the plug, the brood cell, and the brood cell lining. And as you can see on this picture at the bottom right over here, this is kind of how the resin is used in the construction of the nest and the partitioning of the cell. So in some Megachilidae species, resin is used to line the interior of the surface cell, and of course, it's used to construct the partitioning between the cell. So oh, I'm sorry. So resin can also be used, can be mixed with other materials such as gravel, sand, and pebble, as well as organic materials such as leaves, and it act as a binding agent, and it can which can be used to shape or line the nest of the cell. So basically, the resin acts as an adhesive force for the nest. So another thing resin can also be used for is they also act as moisture regulation. So the nests need to be moist enough for the eggs and larvae to develop, but it can't be too much that harmful fungi might grow, of course, and potentially be harmful to the nest. Okay. And so we can actually see this in the long-tongue Megachilidae species and Apidae, where they use the external material like resin in conjunction with surrounding substrate to form thin lining, which is impermeable to water. So another thing that they also use for is they also use it as defense against predator and pathogens. So I want to make a disclaimer here is that we could not find any example in the literature of the usage of resin for defensive purposes by solitary bees, although there are examples for stingless bee. So what I'm about to say afterward, take it with a grain of salt, but I do think it is important to mention. So resin is often used to immobilize predator like ants and beetle. For stingless bee, the resin basically stick to the ants and beetle and stop them from potentially getting into the nest and harming it. So stingless bees has also been observed to self-medicate with plant resin in response to fungal infection, and also in stingless bees, we also found that I'm sorry, there has been they have been observed that during period of increased exposure to harmful spores, they will actually increase the forage rate for resin, which is actually very interesting. So another thing is that resin from different plants is different from one another, of course. So a resin, so a resin from a white pine and a resin from a red cedar will not be the same. So they differ in the chemical composition. Of course, certain compound, certain element might be more prevalent in a red cedar than a white pine than a tamarack, let's say. And they also differ in their utility against different challenges. So of course, the bees face many different threats, many pathogens, many predators, and different resin type is more effective against one threat and less so than the other. Now, next up. So now we're going to look at the solitary bee's life cycle. So first up, first things first is male and female first emerge from hibernation. And then the male and female will mate and they will build a nest, which include collecting supplies such as stone, mud, leaf resin to build it. And afterward, the female lay eggs, with the unfertilized egg becoming male and the fertile eggs becoming female. Next, they will collect the nectar and the pollen, and of course they'll use it to feed themselves, but they also do it. They also leave supplies of food for the eggs. Now, after that, they dies. So the adult stage of the cycle is very short. It's only for a few weeks. And once the parents died, offspring will be left to fend for themselves. Now, the eggs, they will become larvae, of course, and they'll feast off of the food left by the parents. And then the larvae will overwinter in a cocoon. And then, of course, once spring and summer come, they will emerge and the cycle start all over again. Now, again, so the life cycle only takes about a year to complete, and the adult only survive for a few weeks. So this is the observation that I were able to make, and this is the thing that kind of drive my my research topic. So again, so much of my observation is actually obtained from the 2014 paper by Dr. Drescher, where although I will say that her paper looked at stingless bees. So diversity in resin source can be beneficial to stingless bee under a certain condition, of course. So it depends on the targeted organism. So what are the threats that they face? It depends on what the threat is. Like this specific species of ants or this specific species, this specific microbe. And it also depends on the types available, like the resin that I have access to. It also depends on the concentration of the resin as well. So functional use of each resin is helpful against different challenges, as I stated. So this specific resin A, is better against this specific, say, ants than resin B. And maybe against another species of ants, resin B would actually be more effective. So again, different types of resin have varying protective properties, and they're useful against different potential danger that bees might face. Okay, so this is the figure from Dr. Drescher 2014 work. So this look at look at the attraction and repellence of different resin. That is collected by stingless bee against a predator that they might face, which is the meat ants and the green ants, as well as the parasitic beetles that Dr. Drescher looked at in her study. So as you can see here, the resin that Dr. Drescher and her team looked at is the Caribbean pine, the one up top over here, the Cardagi tree, the one right after that, and then the pine. And another thing that I also looked at, that I also compare, is the resin mixture. of them. So first thing first, I want to turn your attention to the first graph, graph A. So this is actually looking at the green ants tests against 5% extract for each of them. So as you can see here, the Caribbean pine, there's actually a slight attraction in that it attracted the green, like the resin actually attracted the green plants. And the same goes, and there's also an attraction when we look at the Cardagi resin tree. But between the Caribbean pine and the Cardagi tree resin, it's not like the point of attraction is not statistically significant. Now, when we look at the hoop pine and the resin mixture from the study, we can actually see that there's not much of an attraction or repellent. And between those two, it's not like they're not statistically significant. Like there's no pointing in either direction. And between the hoop pine and the resin mix, it's their point of attraction and repellent is not statistically significant to the Caribbean pine either. Now, next up, we are going to look at figure B. So this is actually meat ant test against 1.6% extract. So as you can see here, all of the resin, the Caribbean pine, the Cardagi, and the hoop pine, as well as the resin mix, they all show a repellent against the meat ants, but they're not statistically significant. So they're pretty similar to one another in their ability to repel the meat ants. Now, when you look at figure C, graph C, you can see that, again, all of them shows that they are able to repel. Oh, I'm sorry. Graph C over here, you can see that it's a small hive beetle test, again, 1.5%. The small hive beetle test against 5% extract, I'm sorry. So you can see here that the Caribbean pine, the Cardagi, hoop, and the mixed resin, they all show a repellent against a small hive beetle at 5%. But the point of repellent isn't statistically significant between all of them. Now, the interesting thing is when you look at graph D, this is the small hive beetle test again, but this time it's against 1.6% extract. So as you can see here for the Caribbean pine, there's not much of a repellent or attraction either way against them. But when you look at the Cardagi resin, you can see that there is a repellent against the small hive beetle, and the point of repellent is actually statistically significant from the Caribbean pine. And when you look at the hoop pine, you can see there is a repellent, but it's not statistically significant from either the Cardagi or hoop pine. Now, the interesting thing is when you look at the mixed resin in this test, 1.6% extract, you see that there's actually a repellent against the small hive beetle, and you can see that it is statistically significant from the other three resin used. So now we can see that the three, like in graph D, we see that the three pure resin is actually less effective than the mixed resin in this case. Now, so what are we trying to get from this? So my question is, do solitary bees prefer diversity in resin source? So my hypothesis is solitary bees will prefer to collect a diverse range of resin. So this is my justification. If resins are mixed, then the property of the resin types might act together synergistically, such as in the case of the 1.6% extract, mixed extract against small hive beetle, where it was actually more effective than the pure resin. So mixed resin seems to provide similar protection to its pure counterpart for some situations, such as their ability to repel the green and meat ant was pretty similar to the pure resin. And of course, you would need to collect different types of resin in order to be able to have a mixed sample. So So my prediction from this is if solitary bees are offered many different types of resin, then they will prefer to collect from multiple different sources. Now, my independent variable is the types of resin being offered. And my dependent variables that I'll be looking at is the resin visitation. Now, how do we go about approaching this question? So how can we be sure that the data collected actually represent the resin preference and it's not another outside variable that could possibly affect what kind of resin that they go to? So with this question, so we would need to control for factors such as, say, distance from resin source, the floral resources they have that they have access to, and the unique accessibility issues specific to that to the trees. Some resin might produce more. Some resin might be. I'm sorry. Some trees might produce more of this resin, and some tree is simply much harder because of other reason. So from there, we decided on a more control versus a natural approach to studying this topic. Now, what would this look like then? So how we go about this is we decided that we want to do a greenhouse and cage experiments because this would eliminate the factor that I said before, resin source, floral resource, anything that could possibly affect their resin choice. So these are questions to consider for experiments and also how we answer them. So what bees will we be using? So we'll of course be using solitary bees, and it'll be bees that we already collected from last season. So we're not going to go outside and kidnap like a colony or I'm sorry, we're not going to kidnap like a nest of like a solitary bee nest and just take it to our lab. It's already something that we already have access to. So next is what are the resin that we will be offering to them? So the resin, we will be giving them resin from trees that are native to Minnesota, and it has to be large enough in a large enough quantity that we'll actually be able to offer it to the bees. So this is actually one of the challenge. So when we look at conifer trees, of course they differ in the quantity that we are able to collect, of course, but it's not, but it's still enough that we can offer it to them. But let's say if we want to give them resin from temperate angiosperms, then we would have to collect the leaf bud, which doesn't give that much resin. And so if it gives you like, if they give you like 0% resin, if you extract them and they end up giving you like nothing, then of course we can't offer it to the bee. So how do we offer it to them? So we have to offer it in a way that doesn't disturb them and that is easily accessible to them. So we're not just going to be like, we're not going to be taking our hand with the resin and just putting it into the cage and letting them take from it. That is extremely intrusive and will definitely disturb the bees. Oh, yes. Oh, and also, yeah, going back to the third question, how do we offer to them? It has to be in a way that is actually accessible to them. So we didn't exactly just put it on the ground covered by stuff. It needs to be something that that they can easily get to. So how do we actually keep the bees alive? So this is actually the biggest problem that we found in conducting this experiment. So we have to be able to provide all the factors needed to keep them alive in the context of the experiment, of course. So they would need food, of course. They need to have the nectar and the pollen and everything that they need to survive. When and how will we be collecting the data? So spoiler, we would need to find out the best time that they're active and not only that, when they're active and collecting resin, of course. And you'll see later on from our data how we came to the time. Okay, so answer to the previous question posed. Okay, so the first question, what are the bees that we'll be using? We will be using the Megachilidae campanulae. Again, these are the resin that we'll be offering them. We will be offering them the balsam fir, the red cedar, white pine, white spruce, the tamarack, and the red pine, and we were going to offer them mixed resin, but of course, as you will see later on, it did not go. So how do we offer it? How do we offer our resin to the bees? So the resin will be attached to a suspended object. In this case, we use a standard popsicle sticks, which is approximately about 113 millimeter to 9.5 millimeter. And it'll be and it'll be suspended to the top of the cage so that it's easily accessible for the bees. Now, this is the flowers that we'll be using, the creeping bellflower, as you can see on the picture right next to the text here. This is what it looked like, the creeping bellflower. And now, so when is the best time to actually observe them? And spoiler alert, this is actually from our data. We found that the best period is between 15 o'clock to 18 o'clock. So around late afternoon. So this is our cage setup. This is the methods that we use. So first off, we take a large butterfly nest and we turn it inside out so that the top carrying handle is actually inside of the cage. So the reason why we turn the reason why we want the top carrying handle to be inside of the cage is so that we can use it to suspend the bee nest and make it so that there's something that is holding the bee nest up there. So as you can see here, we would suspend the nested handle and we have to make sure it's tight and stable. It can't wiggle around, which could possibly disturb the bees. And we would attach the seating block of the bees to its side. The seating block is of course where the bees will come out and then go to its nest and do all the things that they need to do. So, and then we would be placing the creeping bellflower into the nest. So how did we actually know what to put in there? So the best way that we did it is we just went outside and observed and see which flower was most frequented by the bees. And we saw that the creeping bellflower was observed to be the flower that is most often visited in the wild. I say wild in quotation mark because most of our observation was done on the University of Minnesota St. Paul campus. And this is actually, and as you can see in the picture to the right, this is actually one of the spot that we went to. This is the horticulture display garden of the University of Minnesota St. Paul campus. It has a lot of flowers, so this is the ideal spot to do our observation. But of course, we did it in multiple different places throughout the campus. So this is how we would feed them. So we would feed the bees using a baby feeder jar. So first off, we would poke a sizable hole into the cap of the baby feeder jar, and the baby feeder jar is actually filled with sugar syrup. And then once we got the hole, we can place a short straw into the hole, and we would decorate the end of the straws with petals to resemble the creeping bellflower in the cage. So this is where they can drink out of so essentially the bees can come out and drink from the straws so they can get the sugar syrup and the food, the sugar syrup, which is the food that they need. But alternatively, what we can also do, and this is towards the beginning of our experiment when we were still trying to figure out the best way to feed them. In order to keep them alive, we wet the end of a Q-tip with syrup and we would dab it onto the cage. And essentially, when the bee landed on that exact area, they will be covered in the syrup, and bees would of course try to clean themselves, in which case they will, while they're cleaning themselves, they're also eating the syrup also. Now, so this is our methods for collecting and offering the resin to the bees. So first things first, we would collect resin from, again, the balsam fir, the red cedar, the white pine, white spruce, tamarack, and the red pine. So what we use to collect from them is stainless steel spatula, and we would be using a 4-gram screw Teflon cap vial to collect it from. So this is the cap, and this is the vial in question. I'm sorry about that. So after every collection, we have to make sure we clean the spatula using 70% ethanol to get rid of any polar substance on the spatula, and then acetone to get rid of any non-polar. And we do this after each collection. And of course, the way we collect it from is just we see if there's any resin dripping off of the tree, and then we would just collect them from there. We did not harm any trees in doing this study. One thing you should that I want to know. So after we collect it, we would take portion of each type of resin and we would mix them together and transfer them to a clean vial. So this is our mixed resin that we would want to offer to the bees. So after we got all our samples, we would transfer each of the resin onto a popsicle sticks and we would label them appropriately. So white pine would have, say, WP, white spruce would have WS, tamarack would have T, and so on. So the resin should cover approximately 1/5 to 1/4 of the popsicle sticks. And this goes back to what I was saying before, that the resin need to be in a substantial amount for them to be offered. So using a string and clothes hanging clip, we would suspend the popsicle stick high up into the cage. And after that, we would observe the bees' visitation and collect the data. Okay, so now we're going to preparing the bees. So of course, for this, it's pretty easy. We would just allow the bee to emerge naturally from the seeding block. And in our study specifically, it's around late June. Yours might be a little bit earlier, yours might be a little bit earlier, but for us, we found it's in late June. So once the bee emerged, we would capture it and mark it with a color marker. So, and each individual bee is distinguishable by their unique color. So this bee will be red bee, this bee will be blue bee, and so on. So, and of course, we would collect data on their visitation pattern, including when, what, and who. So when, of course, it's like, what time did they collect it from? What resin did they collect it from? Is it a white pine? Is it a tamarack? So on, and who? Is it yellow bee, blue bee, whatnot? And we actually found that the period. And another thing is that the period for data collection is about 2 hours per day, is how we did it. Now this is the experimental setup. So as you can see on the left picture over here, this is the overall setup of our experiment. So this is the cage, as you can see, the flowers, the popsicle sticks being hung, which you can also see in the right picture over here. And you can see the bee, and you can also see the nest that's suspended by the handle. And yeah. And this is also another thing. So so our nest is made of bamboo. And so as you can see on the picture on the top right over here, this is kind of what it resembles. Of course, it's not like exactly representative of ours, but I think it gets the idea across. Now, if you look at the bottom right over here, you can see this is actually a queen honeybee, but a queen honeybee that is marked, but we use the same technique. The same idea still applies for honeybees. We capture them and mark them with a color. And this is how I distinguish them from one another. So now we actually get to the data itself. So this is going to be the visitation pattern of cage Megachilidae campanulae. So this is all just the raw data. This is what you are seeing right now. Our data was collected from the 27th of June to the 6th of July. And I want you to look at, the thing that I want you to look at is that you can actually see like on our table, there's actually periods where we have none for both resin collected and resin and the resin info and the B info, I'm sorry. So this is actually when we were still trying to figure out when is the best time that they're active in collecting the resin. Of course, and as you go down the data, you can actually see that we actually found the right time, which is during late afternoon when they're collecting resin. So now let's clean up this data a bit. So this table look at the number of visitation for the individual resin. So as you can see here, the resin that is most visited is actually the tamarack at 54 times, and then followed by the white spruce at 25, the red cedar at 16, the balsam fir at 14, and the white pine at 12. So now let's dive let's dive a bit deeper. So table 3 over here, you can see that this is the number of resin visitation for Cage2 yellow bee. So the reason why I want to show you this is because yellow bee is one where we had the most data on. It came out pretty early on, so we were able to collect their visitation pattern over a longer period than the others. So of course you can see here the this Cage2 yellow bee. It visited white pine the most, although we can see here in the blue, the blue is the first day visitation, which you can see that they visited the white spruce and the balsam fir, and the last resin that they visited was on the 6th of July, which is the tamarack. So we didn't see like a strong preference with the yellow bee, but for the next one, it's going to be a little bit different. So this table 4, we're looking at the number of resin visitation for Cage2 blue bee, and this it's the same thing where we did this because we look at Cage2 blue bee because it came out pretty early, so we were able to gather more data for it. So as you can see here, it actually showed a strong preference for the tamarack. It visited 17 times, which is much more than the other resin. That it visited, and as you can see here, much like the yellow bee in the same cage, on the first day it actually collected from the white spruce and the balsam fir, but towards the end it actually started collecting from the red cedar and the tamarack, and it has a very strong preference for the tamarack, even though it's still collected from the others. Now that's all. So that's the data itself. So I just showed you the data, but here are some observations that we generated from the study. So individual bees did, so of course, individual bees did collect from different types of resin over the course of the study. And as I said before, some bees have a clear preference as to what they choose, while others not so much. And we actually found that red cedar and especially the tamarack was actually collected much more often towards the end of the study. There was like it was collected almost non-existent in the first half of our study. But as we go on, we actually see them starting to be collected more often. So we actually found that the bees are most active in collecting pollen during noon to late afternoon. And bees were most active in collecting resin during late afternoon from a period of 15 to 18 o'clock. So resin preferences for resin is again not apparent till a few weeks after emergence. It could be that they're still taking their time to get to be adjusted towards all the resin that's being offered. But again, that's still something that we need to look at. So also another interesting thing that we found is that the bees were actually observed to fight over resin. We actually found that there was 2 bees. They were both trying to collect from one resin source, and they actually fought each other. And the winner, of course, collected the resin, the other went back to its nest. And this behavior is actually also observed in stingless bee, which we also found to be very interesting. So now that we got this, what's next? So the next course of action, the one thing that we weren't able to do in this study and that we were going to do is actually present mixed resin to the bees. I think this is a great way to go about it, of course, uh, since uh, since of course when the bees collected resin, when it get into their nest, it'll of course all be mixed. So I think this is an interesting route to go to, and especially since we saw like the benefit that the mixed resin could possibly have for the bees in Dr. Drescher's. So it would be great to be able to go this route uh, if we were to conduct this studies in the future. So another thing is that we would want to introduce resin from non-conifer trees. So as I already said before, it's kind of hard to collect enough, but if there's a way to collect more to collect and be to collect in a large enough quantity and give it to them, it would be a great way to go, because in the natural setting, bees would of course collect from non-conifer tree resin as well. So again, we would perform the experiment over a longer period of time. We did not have that many days to collect data. And as I already said before, uh, preference isn't very strong until later on. So if we were to have enough time, say about 2 months, we think it would yield better results or better data, I'm sorry. And again, we would want a uniform observation period between the days. Now that we observe that the bees come out during late afternoon, perhaps we can focus on that instead, so that we can get like good data, so that we don't have like several days of like nothing. Another thing that we can also do is that we can also analyze the chemical composition of the resin. So we would want to know, like, if they do prefer this, or if they don't prefer this, what is it about it that they like, or what is it that they don't like? And we can relate this results, say if we run a GCMS or LCMS, we can relate this possible result to the result from the study that we generate. Like if they have a preference for, say, red cedar, what kind of things about red cedar that they actually like about them? And again, we would want to set up a natural experiment to test out this hypothesis. So this is a pretty ideal scenario where nothing else is really interfering. It's really only resin preference that they're going after, because for resources given to them, that's not a factor, and a lot of other things is eliminated. But of course, in the ideal world, that's not how it works. So again, we would set up a natural experiment to test out in conjunction with our more controlled. Now that we did all this, what is actually the application of this study? So of course, information gathered from this study about solitary bee rearing can help develop methods for other lab-based experiments. So I want to emphasize that there's not much study, or at the very least, I was not able to find any study that actually talked about rearing solitary bees. So all this information that I did came up by me and my team. This wasn't gathered by the information was not gathered from literature. So the information that we gathered from this study can be very helpful for other experiments that want lab-based experiments, that want to rear solitary bees. Another thing is that the result that we gather can also help focus in onto resin that is specifically used by solitary bees. If for example, a white pine is not even touched by solitary bees, then it might not be as useful to study them as opposed to a red cedar that they always go to. Well, this is the end. So I want to give a special shout out. So special thanks to Maddie Oliver, Julia Grabo, Ethan Ravela for their help in carrying out the project. And special thanks to Dr. Michael Wilson for his support and guidance throughout this whole project. Special thanks to Thea Evan and Joe Holdridge for their help during the data collection process. I really appreciate their help. I really appreciate their help in being out in the very hot sun and helping me collect the data. And special thanks to Dr. Jerry Cohen for the use of his lab and knowledge on the St. Paul campus. And also special thanks to Christine Solomon for the use of her lab. And yeah, this is a list of my source. I hope you enjoy my presentation. Yeah, well, this is it. This is my research. Bye.